of disease that affect motor neurons or nerve cells in brain or spinal cord that will tell the muscles what to do. These diseases affect both the adult and children. These diseases affect the patients differently, but they all show movement-related symptoms.
Some motor neuron diseases are inherited, but the cause of most motor neuron diseases are unknown. In sporadic or non-inherited motor neuron diseases, factors like toxic, environmental, viral, and genetic may be implicated.
There is no single diagnosis test for motor neuron diseases. Diagnosis tests usually based on features in the clinical history and examinations, which includes expensive techniques like DTI and MRI scanning.
These techniques are only available in multi-specialty hospitals and medicities. In a developing country like India, these facilities are not affordable and not available to every person. We, the students of Amrita School of Biotechnology, as part of a final year BRIGHT project, are conducting further research on finding cost-effective methods for the diagnosis of motor neuron diseases.
Electroencephalography is a non-invasive technique used to detect the electrical activity of the brain. Compared to other neuroimaging techniques, EEG is inexpensive and can be used to detect the abnormalities in the brain signals for the mode of walking or gait patterns.
For our experiment, we are using a 14-channel device positioned according to the 1020 International System. The felt parts are dipped in saline for better conductivity and placed in the electrodes. These electrodes are then slotted into the EEG device. Open consent is taken from the participants prior to the accelerometer and EEG recordings. The device is placed on the scalp and arranged so there is a 3-finger gap between the brow and the electrode, and so that the electrodes are touching the scalp. The EEG is standardized before the start of the experiment. The participants are given instructions to avoid eye blinking, swallowing, or any other unnecessary movements. They are asked to keep their gaze straight ahead and trained in the task before the start of the experimental recording. There is a 30-second relaxation period, after which a vocal command is issued to walk. After the completion of 4 gait cycles, the recording is stopped.
We have implemented 2 sets of protocol in our experiment. The first set of protocol has EEG placed on the subject, while the second set of protocol has EEG as well as accelerometer strapped onto the subject's lower body. Accelerometer is an application that is available in all app store for Android phones. It measures acceleration as well as angle values at the joints where it is attached.
In our second protocol, along with EEG device, accelerometer sensors are also used. They are strapped to the lower joints such as hip, knee, and ankle on both the sides. Recordings are started in EEG as well as accelerometer. After a 30-second relaxation period, a vocal command is issued to walk. After the completion of 4 gait cycles, the recording of accelerometer is stopped and then EEG. The acquisized data is pre-processed, extracted, and analyzed using various tools. In the results we've obtained through spectral analysis, we observed that alpha band is comparatively higher and more prominent in the parieto-central regions, followed by theta in the occipital regions and beta in the anterior frontal regions.
When both these sensors are used together, it can further validate the diagnosis or give a clear picture in the case of unsure diagnosis. In closing, our project contributes to paving the way for further research in providing accurate and low-cost diagnosis so it can be made even to economically backward society.